In this orientation, only had interaction with serine 280 and phenylalanine 251, which probably explains why it's a weaker PPAR alpha agonist compared to pterostilbene. Having seen this good activity of pterostilbene, I decided to, being with the U.S. Department of Agriculture, I decided to analyze several fruits, thinking that by finding a fruit which has high levels of this compound, you could directly benefit from its effects by consuming the fruit. So I studied a lot of different compounds, analyzed it, but I studied strawberry, cranberry, I think I even studied corn, but I found it only in species of vaccinium as listed here, arboretum, SGI, that's rabbiti, high bush, Elliott's blueberry, and deerberry. And I've got collaborators who gave me samples from all over, just to show how I monitored pterostilbene in the samples. This is by GCMS. These are the standard compounds, and that's resveratrol right there, and this is pterostilbene. And doing this by derivatizing pterostilbene and resveratrol, monitoring for 4,4,4-mess for resveratrol and 328 for pterostilbene. And doing that, we were able to show the levels of pterostilbene in some of these fruits. The highest level was found in one sample of high bush blueberry, and I believe, as far as I remember, this is a sample provided by a collaborator from Agri-Food Canada. So even of the same species, high bush, 3 different samples have various levels, variable levels of the compound. So I presented this research results at a meeting, like we should do, as required of our job. Who knows, faculty members know about that. Even the students are required to present their work. And this was presented at the American Chemical Society ACS National Meeting in Philadelphia in 2004. And to my surprise, this paper was selected by ACS as newsworthy. That I don't even understand why they selected it, but I'm happy for that. Anyway, so what happened thereafter is me being busy running away from reporters. Because actually, this is the website of the High Bush Blueberry Council way back in 2004, where they unbelievably catalog all of the news items that came out of my work. And these are just a few newspapers and television and radio stations that broadcast my work. That's why I said I couldn't hide. They find me even my hotel. I don't know how they got the number of my hotel. And that's just one. And they also, that's in the US, and they were also broadcast in Canada, China, Russia, all over, India, what's that, Netherlands, United Kingdom, South Africa, and there's a lot more even actually in the Philippines. My mother was surprised. And she practically bought the newspaper where this was reported. And I was surprised too that I didn't know it was even broadcast in the Philippines. But this is the one I like the most, BBC, and also a newspaper in UK, the most widely distributed newspaper, the London Independent. Because as you can see there, they say in Tesco, the largest supermarket in Britain, I couldn't read this very well. Largest supermarket in UK, there's 185% increase in blueberry sales. And even at another supermarket at Walrose, there's 150% increase in blueberry sales. And this was, the main stimulus is I don't have my glasses, a report last year by a researcher from USDA. It showed that pterostilbene could be as good as a commercial compound for lowering cholesterol. And that I showed in my data, experiments from my data. So these are just some of the examples of the news release. It's considered one of the top 5 best food news by Parade Magazine in 2004. So there it is. I just highlighted that one, or exploded USDA laboratories in Oxford, Mississippi found pterostilbene. And it also made the Time magazine cover in 2004. Unfortunately, they say blueberries, but not pterostilbene, or not my name, but I know that was my word. Yeah. More than that, beyond that, then there are other new studies that we did spurred by this first successful research. And so had several people call me, wanted to work on my compound, and so we had a lot of studies that show its effect on obesity. We have shown that already and also we have studies that show it lowers blood pressure. This is just a slide of metabolic syndrome, a cluster of cardiovascular risk factors. So I just want to say that my compound has positive effects on some of these components of the metabolic syndrome. So this is the study in rats fed 2 doses of pterostilbene at 15 milligram per kilogram body weight and at 30 milligram per kilogram body weight, where it shows total fat decreases. This is control, and this is a PT15 group, and see the decreases in fat depots. And we look at the mechanism of action of how it controls obesity, and just a simple scheme for lipogenesis being the process by which acetyl-CoA from metabolism of simple sugars that we eat gets converted into fats. So the process of glycolysis, glucose gets broken down into C3 pyruvate, which enters into the citric acid cycle. The citrate from that metabolism goes out of the cell and then gets converted into fats, and the fat esterifies with glycerol to get triglycerides. And increased synthesis of triglycerides, of course, causes obesity. And pterostilbene works also not just by inhibiting all of these enzymes indicated here that inhibits fatty acid or lipogenesis, but also increases fatty acid oxidation, and this, as I said, is due to PPAR alpha activation. So PPAR alpha, the target genes are these genes of the carnitine palmitoyl-CoA transferase and acyl-CoA oxidase, which are the committed enzymes for fatty acid oxidation. More readings if you're interested. This has been reported already, so I can refer you to the publication later. But this is a clinical trial that showed that high-dose pterostilbene at 250 milligram per day significantly reduced blood pressure compared to placebo. And also some clinical safety trials done. It was shown that pterostilbene had no adverse effects on hepatic renal glucose markers, and it is generally safe up to a dose of 250 milligrams per day. In addition to the effects on the metabolic syndrome, pterostilbene also is found, or we found, to have beneficial effects on mind function. And one of the studies, so this is just to say that pterostilbene also is found in the brain, not just in the liver, where I said it's predominantly found in the liver. And the activity, or at least its effect on the brain, is not on the lipid synthesis, but is to decrease oxidative stress in the brain. And it's also found in the heart. We haven't had any study to see the effect on the heart yet, but I'll be showing the next few slides the effect of pterostilbene on oxidative stress in the brain that affects memory and cognition. So this is one of the first studies we have where we used cells COS72 cell, which is transfected with a muscarinic M1 gene, which is the most vulnerable to oxidative stress. So the way it is here, this is the normal cell, which is not treated with dopamine, which is the stressor in this situation. And then once it is treated with the stressor, dopamine, then the cell does not recover from releasing calcium. There's membrane dysfunction due to the stressor. But here, positive control use was blueberry. And you can see that with treatment of blueberry extract, the effect of the stressor was negated. So still the cell functioned like the cell not treated with the stressor. And among the stilbenes tested in this set of compounds, as I see that there is stilbene and glycosylated pterostilbene, which I synthesized, are the two compounds that behave like the blueberry, which protected the cells from the stressor, from oxidative stress. So we did animal study to kind of to show really if it will function in whole living system, because that was a cell assay. And this is already reported in Journal Agricultural Food Chemistry in 2008, where we used the Morris water maze test, which involves placing the rodent in a pool of water where it must use external cues in the testing room to find a hidden platform just below the surface. And this measures spatial memory and learning. So what happens is, at trial 1, you put the mouse or the rat in one starting point, take them out just to allow them to navigate and get familiar with the room until they learn where the hidden platform is. And you take them out overnight, make them rest, and the second day, you put them back at the same position. And the rat, which has good memory and competent memory functioning, will be able to know, based on cues around the room, to go directly to the hidden pool. Whereas a mouse, which has malfunctioning memory, then will have to have longer navigation to be able to get to the hidden platform. So given this model, we treated the mice with 2 doses of pterostilbene 1 and a low dose we